Okay, welcome to stage 2 of protein synthesis, translation. If you haven't watched the introduction to protein synthesis video, click the link up here and watch that one first, and then watch the stage 1 or transcription video prior to watching this one. In our last video on transcription, we transcribed the sense side of the messenger of the DNA into a strand of messenger RNA. We edited that message down, cutting out the introns, splicing together the exons. We put a methyl cap on the 5' end and a poly A tail on the 3' end. And our mature transcript was ready to leave the nucleus to go to the ribosome for translation. And that's where we're at now. Translation, the process by which messenger RNA information is changed into the actual protein at the ribosome with the help of transfer RNA. Let's take a closer look at ribosomes. Ribosomes are the organelles of protein synthesis. They're small and found in the cytoplasm, sometimes attached to the endoplasmic reticulum, sometimes free-floating. They're made of both protein and ribosomal RNA and divided into two subunits. If we look at this picture, it's easier for us to see the subunits. We have the large subunit on top and the small subunit on the bottom. This picture is also showing the messenger RNA and transfer RNA already loaded, but we'll get to that in just a moment. When we look at translation, it's best to look at it in 3 steps. The first step is initiation, where the messenger RNA strand binds to the small subunit of the ribosome. Then, the first transfer RNA comes in and lines up. At one end of the transfer RNA is a 3-letter code, which we call an anticodon, which is complementary to the corresponding codon on the messenger RNA strand, which is coding for a specific amino acid. At this end of the transfer RNA, we have an attachment site for that specific amino acid, in this case, methionine. And now we're ready for the last part of initiation, where the large subunit comes in and binds, finishing our initiation complex. The large subunit has a tunnel that the messenger RNA can travel through. Actually, the ribosome is going to travel down the messenger RNA, kind of like a train down a track. And we can see that our first transfer RNA carrying methionine is parked in the P binding site. We have 3 binding sites, the E site here, which is going to be our exit, the P site in the middle, and the A site, which one of my students suggested we call the acceptor site, which will make sense in just a moment. And now we're ready for step 2 of translation, elongation. The next transfer RNA will come in and park in the A site, or the acceptor site, and lining its anticodon against the codon, and we can build a bond between these 2 amino acids. If we recall, that bond is a peptide bond, and the ribosome acts as an enzyme, help catalyzing that reaction. So we've made the first link in our amino acid chain. And now the ribosome slides over one reading frame. This first transfer RNA is now in the E site, so it can exit, and the next transfer RNA can move into the A site, lining up its anticodon next to the corresponding codon in the messenger RNA chain. We build another peptide bond, and we are elongating our amino acid chain. So we can continue. The ribosome slides over another reading frame. The transfer RNA exits as the next transfer RNA comes in, and we build our next peptide bond. And you can see how this is going to progress until we get to this reading frame. And now you'll notice that we've reached a codon, UGA, which is one of our 3 termination codons, without a corresponding amino acid. This is the signal to the ribosome that it's done translating this amino acid or this messenger RNA strand, which means we're at step 3, termination. Now the components of the initiation complex, the large and small subunit, are going to disengage and release the amino acid sequence that can go on to take its final 3-dimensional shape. Now in this case we made a very short amino acid strand of only 4 amino acids, a very small protein, but imagine this to be 100 or 200 amino acids long. And we're seeing it in a very linear way here, but we know that proteins will twist and fold and kink into complex 3-dimensional shapes as it takes on its secondary and tertiary structure based on hydrogen bonding and R group interaction, but that's for another video. Now these two ribosome components can go on to reform, maybe go back to the beginning and start to translate this messenger RNA codon a second time to make, or this messenger RNA strand a second time to make another copy of this protein. And this messenger RNA strand can be translated many times by many ribosomes. In fact, it's not uncommon for one messenger RNA strand to have more than one ribosome translating it at a time. It's called a polysome. We can see it in this picture. This orange line down here represents our messenger RNA strand, and the ribosomes are moving down like trains on a track, one after the other, moving from the left to the right here. And so this is one that's reached termination and is splitting up, and this one's at the very beginning. And you can see as they travel along, they're extending this amino acid sequence. And this is an amazing photograph down here. You can see with electron microscope, we can see the messenger RNA strand here, and the dark dots that are along it are individual ribosomes that are traveling down the track, and you can even see the proteins kind of coiling off of the ribosomes as they're being constructed as the ribosomes move down the track here. It's a pretty amazing photograph, I think. And again, when we have these multiple ribosomes attached to one messenger RNA strand, we can call that a polysome. Okay, so that brings us to the end of the details of protein synthesis, transcription, and translation. And in this step, we took our messenger RNA to the amino acid sequence at the ribosomes with the help of transRNA. So if you need to, go back and watch the previous videos, but this ends the protein synthesis section of this unit.